And we said that membranes are not static, right? So they carry out functions and they're asymmetric. This asymmetry helps you determine, which way different molecules move or which way different signals are received in different students. All right, so let's talk about that today. We're going to talk about what's going on in the membrane, right? What actions do they perform? And one of the main things that membranes do is, of course, they separate the inside of the cell from the outside of the cell, okay? And in order to move molecules in and out, right, so we'll start talking about molecules first, is using pumps, channels, and carriers, okay? So pumps and carriers are examples of what we call active transport. Active transport requires an input of energy because we're going to be moving things against their concentration gradients. You can imagine we have a hypothetical hole in the membrane. The molecule is going to want to move from the compartment with higher concentration to the compartment with lower concentration, right? If we want to move it the other way around, from an area of low concentration to an area of high concentration, well, that's going to require energy because that's thermodynamically less favorable. So this is what active transport does. In the case of pumps, it uses energy from ATP, right? So this is straightforward. We know that ATP hydrolysis can provide energy to a process or reaction, and that's what's going on in a pump. We're going to go over some examples of pumps in just a minute here. More interesting, though, is carriers. Right? Carriers use energy in a different way. They don't use energy from ATP hydrolysis. Instead, they use energy in a much more interesting way that we'll see in the next couple slides here. Channels are an example of what we call passive transport. That's the example I gave just a moment ago, right? Some pore that's open in the membrane and selective for a specific molecule or ion, but this simply just allows that molecule or ion to move down its concentration gradient. And so what we see is that these active transporters and passive transporters are in a way paired, right? Because in order for a molecule to have a concentration gradient, that concentration gradient must have been established at some point, right? So an active transporter establishes a concentration gradient, and that concentration gradient can pass through a channel without any input of energy. Right? We'll see that when we talk about action potentials. So carriers again, right? They use energy in a different way that isn't ATP hydrolysis. And what energy is that? Well, membranes are any two compartments separated, right? They have a free energy, which is to say if we have a concentration of 1 molar on one side and a concentration of 0.1 molar on the other side, well, of course, right, just intuitively, you know that the molecules are going to want to move from the compartment that has 1 molar to the compartment that has 0.1 molar. And it's going to happen spontaneously, right? And what do we know about any process that occurs spontaneously? It must release energy. The Gibbs free energy for that process or reaction, in this case process, must be negative. And we can actually quantify the amount of potential energy that's stored in the system that is made up of a concentration gradient. Delta G, right, that potential energy, that Gibbs free energy is equal to R, the gas constant, times T, the temperature in Kelvin, times the natural log of C2 over C1. Okay? And what does this tell us? Well, C2 and C1, we often just use C final and C initial. Right? So this is the concentration of the compartment in which we're going to go. And C1, C initial, is the concentration in the compartment that we started with. Right? So if we're moving from the outside of the cell to the inside of the cell, in this case C2 would be the concentration on the inside of the cell, and C1 would be the concentration on the outside of the cell. And we can see that actually just by flipping it, all we do is reverse the sign of this term, right? So we just, it's the same magnitude, just the opposite sign, right? So depending on which way we're moving, now we see that we'll either have a positive delta G or a negative delta G. And for a process to be spontaneous, well, we'd have to be moving from an area of high concentration to an area of low concentration. Conversely, if we want to move from an area of low concentration to high concentration, all we're doing is flipping this term inside the log, so we take the negative. So now instead of releasing that amount of energy, we have to put in that amount of energy to establish that concentration gradient. Okay? Now this is true when we have an uncharged molecule. Okay? When we have an uncharged molecule, the only thing driving this process or requiring an input of energy to establish this concentration gradient is simply the concentration of the molecules. But what happens when we have a charged molecule? Right? So for various reasons, we're going to encounter these today, right? We have to take into account the charge on a molecule. So the full equation is the same, delta G equals RT ln C2 over C1 plus ZF delta V, right? Now ZF delta V tells us the electrical gradient across that membrane. Okay, Z is simply the electronic charge. So on a sodium ion, for example, C would be just positive 1. On a chlorine ion, it would be negative 1. F is Faraday's constant. You should commit this to memory, just like the gas constant, 96.5 kilojoules per mole volt. And delta V is the change in electrical potential between the two compartments. Okay? And this is actually relevant for a cell. If we consider we have a cell, okay, cells are not electrically neutral. Okay? They actually have a membrane potential dictated by the concentrations of a bunch of different ions. They're VEQs. We'll see how to calculate VEQ later on today. But the membrane potential is equal to V in minus V out. Okay? So what does this mean? That means that it's equal to the difference in electrical potential between the inside of the cell and the outside of the cell. This typically is on the order of negative 60 millivolts. Okay? And of course, because this is V in minus V out, we can always assume that the outside of the cell is 0 volts and the inside of the cell is negative 60 millivolts. Now we have negative 60 minus 0. Okay? It's the easiest way to do it. Why do we need to know the membrane potential? Because now we can fill in this delta V term. Okay? Let's assume again that we have sodium ions at some crazy number, let's say 500 millimolar on the outside, and we have concentration of 10 millimolar on the inside. Okay? If we were to punch a hole in here, okay, how much energy would be released? Okay, how much energy would be released by the sodium entering the cell? Right? I might ask you that. So for this, all we have to do is say that delta G equals RT ln, right? So what's our C2 going to be? Well, it has to be 10 millimolar or 500 millimolar, which is C2. 10, right? 10 millimolar. Doesn't matter what the units are as long as they're the same. So here we have millimolar and millimolar, so we're good. So C final 10 millimolar over 500 plus Z, which in this case is just 1, so 96.5 times delta V. Now delta V is different than membrane potential. This is where a lot of people get tripped up. Membrane potential is always V in minus V out. Delta V is again V final minus V initial. So in this case, V final is negative 60, V initial is 0, right? So negative 60 minus 0 gives us negative 0.06 volts, right? Important to convert it to volts because notice the units on Faraday's constant has volts on the denominator, so we need to convert our millivolts to volts. And now, right, what is our delta G going to be? Well, this term in here is less than 1, which means it will be negative, right? So let's just say it's negative something plus, in this case also, this term will be negative, so plus a negative, so delta G will be quite negative. And this makes sense intuitively again because we have a positive ion moving into an area of low electrical potential or negative electrical potential, so that's favorable. We have going also from high concentration to low concentration, so both of these are favorable, both of these terms are negative. Delta G then will be negative. Conversely, right, how much energy is required to establish this concentration gradient? Well, now we just reverse everything, right? Now we say we're moving in the opposite direction. So we flip these, which means we reverse the sign, so this becomes positive. Here, now we're going 0 minus negative 60 millivolts, which is positive 60 millivolts. So again, we're simply reversing the sign here. So notice that the delta G term will be of the same magnitude, but simply in the opposite sign. So we can either release that amount of energy by allowing it to move freely, or it will require that amount of energy to establish this concentration gradient. Okay, just depends on which way the molecules are moving. And so we see that there is energy stored then in a concentration gradient, right? So these concentration gradients are essentially like batteries. They're holding on to energy. These concentration gradients are used by the cell all over the place in order to power different reactions. We're going to see this in chapter 18 when we discuss oxidative phosphorylation. We're going to see this today when we talk about carriers. Carriers use the energy stored in a concentration gradient of, say, molecule A to power the movement of molecule B against its concentration gradient. So they're exchanging the potential energy of one concentration gradient to establish a second concentration gradient. All right, so let's start with pumps. Pumps are a little bit easier to conceptualize because here we're moving things against their concentration gradients. Of course, this requires the input of energy, but in this case, the energy is going to come from everybody's favorite molecule, ATP, right? So we have some examples of these ATP-dependent pumps, such as the sodium-potassium ATPase. Okay, we'll see this actually in a couple of slides. And this actually establishes the sodium-potassium gradient in cells, which helps to contribute to this membrane potential of negative 60 millivolts, the resting potential. It also is responsible for generating these concentration gradients of sodium and potassium, which allows neuronal signals to propagate down the neuron, which we'll see later today. Calcium, the calcium ATPase, also known as SERPA, the sarcoplasmic and endoplasmic reticulum calcium ATPase. This is responsible for calcium sequestration in the lumen of the sarcoplasmic reticulum, and for a reason that we'll see here in just a minute. And the proton potassium pump. Okay, this is what actually generates that low pH that we find in gastric juice for stomachs. Right, protons are pumped out of the cell into the lumen of the stomach, and potassium is pumped in. Right, so remember the pH of the stomach is about 1, so the concentration of protons are really quite high. Okay, but we use the concentration gradient of potassium to move protons in a way, right? But it's also an ATP dependent pump. Okay, so now let's look at specifically P-type ATPases. And P-type ATPases are named for the phosphorylaspartate that is formed upon ATP hydrolysis. So what we have effectively is a group transfer reaction where the gamma phosphoryl group of ATP is transferred onto the C-terminus here of an aspartate, right? So we have this phosphorylaspartate intermediate, okay? And this helps to change the configuration of our P-type ATPase, okay? Because essentially what we want is a pump that is open to one compartment and closed to the other compartment in order to bind something. Then we input some energy, again here in the form of ATP, and this is going to cause the part that was open to close and the part that was closed to open, so now this molecule can be released onto the other side of the membrane, right? Remember, we can't just have molecules diffusing freely through the membranes, especially in the case of these charged ions like sodium, potassium, and calcium. They're impermeable to the membrane like we talked about last week. So here we see the structure of specifically SERPA, the sarcoplasmic and endoplasmic reticulum calcium ATPase. And we see that it's got 4 different domains, right? We have a transmembrane domain. This is what's embedded into the endoplasmic reticulum membrane. Okay, we have our A domain, our P domain, and our N domain. And we'll cover what each of these do in turn. Where we see the aspartate that is going to be phosphorylated to form our phosphorylaspartate intermediate is in the P domain. That's what the P stands for, phosphoryl domain. The N stands for the nucleotide binding domain, right? This is what's going to bind ATP. And the A domain stands for the actuator domain. So once ATP binds in the N domain, the gamma phosphoryl group is transferred onto aspartate 351 in the P domain, generating phosphorylaspartate. This causes a conformational change in the A domain, which closes the channel that was open and opens the channel that was closed in the transmembrane domain. So all 4 of these components are going to be working together. And what the SERPA pump does is essentially move calcium from the cytosol into the lumen of the endoplasmic or sarcoplasmic reticulum. Reason being that in the cytosol, high enough concentrations of calcium tend to precipitate, right? They form this insoluble precipitate, and this damages the cell physically. So calcium cannot be present at high concentrations in the cytosol, so where does it put it? It puts it in the attic, basically. It just shoves it out into the endoplasmic reticulum lumen, and there it can remain. So the lumen of the ER or the SR muscle is simply just, well, among other things, obviously, but in this case, it's simply just holding on to excess calcium. And why do we need to hold on to excess calcium? Right? Well, we'll talk about in chapter 14, calcium release from the sarcoplasmic endoplasmic reticulum into the cytosol triggers muscle contraction, right? And we can't have muscle contraction unless there's calcium in the lumen of the ER or SR in order to come out into the cytosol, right? So this circle pump is critical for maintaining the proper calcium concentration so that muscle contraction can be triggered, right? So here's the schematic representation of how the circle pump works, right? So again, we have the A, B, N domain, and we have our transmembrane domain, which currently is open to the cytosolic side. Okay, so it's going to bind two calciums on the cytosolic side. You can't bind any calcium on the luminal side because it's close to the luminal side, even though the concentration of calcium is higher here. Okay, so we're moving calcium from an area of low concentration in the cytosol to where it's already high in the lumen of the ER. ATP binds into the N domain, the nucleotide domain, right? And we call this the E1 calcium 2, right? So E1, because we're in the first configuration, bound to two calciums and bound to ATP. Now we get a group transfer reaction from, again, the gamma phosphoryl group, the last phosphoryl group on ATP, onto aspartate 351 to form our phosphoryl aspartate intermediate. We call this the E1P calcium 2 ADP complex. Right? E1, we're still in the first configuration because we're open to the cytosolic side. P, because the enzyme has been phosphorylated. Ca2 plus 2, we're holding on to two calciums, and now we're holding on to ADP. What happens now? Right? Well, the ADP is now released. Okay? And when the phosphoryl aspartate is formed, this causes a conformational shift in the actuator domain, which actuates a shift in the transmembrane domain, which closes this open bit here and opens this side, right? Now this calcium is free to leave into the lumen of the ER, right? This conformational change also decreases the affinity of the transmembrane domain for calcium, so it's easier for the calcium to leave into this area of higher calcium concentration. And now we're in the E2P conformation because we're in the second configuration of this enzyme, but we're still phosphorylated. In order to reset this enzyme, all we have to do now is allow the spontaneous hydrolysis of this phosphoryl aspartate, releasing this inorganic phosphate, restoring phosphoryl aspartate to just aspartate. Okay, now we're in the E2 configuration because we're still in the second configuration where this is open and this is closed. Now, with the release of this inorganic phosphate, there's no longer any pressure on this A domain to be as it was here, so this reverts to the way it was, which of course actuates this back to the way it was, and now we're in the E1 configuration. Okay, we're back in the original configuration, ready to bind two more calciums from the cytosolic side, and resetting the entire system back to the E1 calcium 2. Right, so this proceeds over and over and over and over. And it's the donation, or the group transfer of the phosphoryl group from ADP onto this aspartate that drives this conformational change, that closes the cytosolic side and opens the luminal side so that calcium always moves out in this direction. And again, an important thing to remember is here, if the affinity of this circle pump for calcium wasn't decreased in this case, well, what would happen? Yeah, maybe these calciums would leave, okay, but because the concentration of calcium is so high inside, well, we just get two more calciums binding. And then once we revert, we would be moving those calciums back into the cytosol, and all this process would be for naught. An important part of these ATP-dependent compounds, specifically P-type ATPases, is that upon the switching of what's open to what's closed and what's closed to what's open, the affinity for the ligand also decreases to ensure movement mainly in one direction. Okay? And actually, this is an interesting story. One of my pet interests is medieval medicine, and here we have foxglove. Okay? Foxglove contains a high amount of this compound here, wabane. Where it is. It's in the main note on this slide. But here, this wabane actually doesn't really affect circle pump at all. What it does affect is another P-type ATPase, specifically the sodium-potassium pump, right? And how does wabane work? And what does it do? Well, wabane was shown, or foxglove. They didn't know what wabane was, but foxglove was used to make kind of a primitive medicine, and it was fixing this condition that they used to call dropsy, where people would just drop dead. Didn't know what it was, but one of these medicines involved foxglove, and it seemed to help. Now that we know what dropsy is, effectively it's just the inability of the heart to pump strongly enough, right? And of course, the heart is mostly muscle, and what do we need in order to trigger muscle contraction? Well, we need calcium in the cytosol. It turns out that this condition is caused by decreased levels of calcium in the cytosol. So in order to combat this, right, well, what we can do is try to increase the amount of calcium in the cytosol, right? And that allows the heart to pump faster, and it does help this condition. And wabane here, which we find in foxglove, inhibits this sodium-potassium pump. The sodium-potassium pump is another type of P-type ATPase, and this is responsible for moving potassium into the cell and sodium out of the cell, right? Now, this doesn't have anything to do with calcium yet. We have to remember a lot of these ATPases, a lot of these pumps actually work together in synchronization. So when we inhibit this sodium-potassium pump, what happens? Well, we're not moving sodium out and we're not moving potassium in. Okay, so the level of potassium on the inside will be lower and the amount of sodium on the outside will also be lower because we're inhibiting this pump. So next on we have this sodium-calcium exchanger. So this is a type of secondary active transport we'll talk about later. This uses the concentration gradient in sodium, right, to move calcium out of the cell, right? So here we're going to be moving sodium in and calcium out. By inhibiting the sodium-potassium pump with wabane, right? Well, now we have lower calcium here, which means there's less energy available to move calcium out, which means that calcium would be higher on the inside than it would otherwise be. So now we've effectively increased the concentration of calcium inside the cell, allowing muscle contraction to be triggered more forcefully. And this counteracts the effects that lead to this condition, dropsy. We also have ATP binding cassettes, ABC transporters, right? And these also use the energy of ATP to move molecules against their concentration gradient. We have two examples here: multi-drug resistance protein, which is expressed in bacteria, and this actually helps to confer resistance to different antibiotics, right? So in this case, what they're moving out of the cell is antibiotics. And we can imagine, why would this be useful and why do we need energy for this? Well, let's say that we had, a culture of bacteria to which we add antibiotic. Well, now the concentration of antibiotic in the cell culture media, right, their food is going to be high. The concentration on their inside initially is low, right, and they want to keep it low. So any antibiotic that does get into the cell, they want to move it out of the cell against its concentration gradient. This requires energy, and this multi-drug resistance protein is one of the proteins that allows that to happen, right? It uses energy from ATP hydrolysis to move antibiotics back out, thus conferring resistance to that antibiotic. We also have bacterial lipid transporters. So this is responsible for moving certain lipids from the inner leaflet to the outer leaflet. Okay, but let's just look at the structure here. In the multi-drug resistance transporter, what we have are 4 domains in one polypeptide. We have 2 ATP binding cassettes, or regions, right? So this is where ATP is going to be bound. We're going to bind 2 ATP molecules, and we have 2 transmembrane domains, right? Membrane spanning domains here in yellow. In this bacterial lipid transporter, which is another type of ABC transporter, ATP binding cassette, right? We have a dimer made up of 2 polypeptide chains, but each monomer has an ATP binding domain and a transmembrane domain. And they form these dimers so that they look functionally the same as if we had 4 domains on one polypeptide.
Does the drug-resistant protein happen through evolution?
Huh?
Does it happen through evolution? Like, how they say, when you take antibiotics, eventually they stop working because.
Sure. Yeah. So it's a little more complicated than that. Okay. So we can imagine 2 scenarios. Either we have a population of bacteria where only 0.1% of those are resistant to the antibiotic, right? If you don't finish that entire course of antibiotics, right, well, now you're giving that 0.1% of bacteria, you're giving them a chance to represent more of the population, right? And after a while, now you'll have, 50 to 99% that drug-resistant bacteria. Additionally, right, bacteria are able to share information in the form of plasmids. We've talked about plasmids before. This is one of the ways that antibiotic resistance can actually be conferred from bacteria to bacteria. They can share their plasmids, which may be containing one of these MDR proteins, and now bacteria that were susceptible to this antibiotic now become resistant to the antibiotic. So the trick with antibiotics is finishing the course quickly enough, right, with enough antibiotic so that you don't give a chance for that 0.1% to make up a larger percentage and to kill all those that are resistant or that are susceptible to it. And now all those tiny little amounts that are left, right, those can be easily handled by the immune system, right? That's a good question. All right, and so how do these ABC transporters work? Well, here in this case, we are simply hydrolyzing ATP, right? And this is causing what we call an eversion event. So here what we have, for example, this is the lipid transporter, MSBA, from bacteria. And what we have, right, again, are the two ATP binding sets and the two membrane spanning domains. Each of these is going to bind an ATP, right? And what happens, right? So we bind our ATP, the ATP binding here, the ATP binding causes an eversion event, and in order to revert back to the original state, we have to hydrolyze the ATP. So let's go through each of these steps one at a time, right? So we have our initial state, which is open to the cell interior, okay? And we'll get binding of our ligand into the transmembrane domain. Again, in our initial state, we have high affinity for our ligand. Now we are in the open to the cytosol, closed to the outside state. Next, we get binding of ATP to each of the ATP binding cassettes, and this causes the eversion, right? Causes the two ATP binding domains to come closer together, as we see over here, right? And when that happens, it causes these two sides to move farther apart on the top, thus releasing the ligand, also decreasing the affinity of this ABC transporter for the ligand so that it can be released, right? So now we have the ligand on the outside. In order to reset, we have to undo this eversion, right? And what's causing these two regions to move together is the ATP itself. But when the ATP is hydrolyzed, there's no longer any attractive force between the ATP binding cassettes, so these are able to move farther apart, and these two move closer together, resetting the entire system here, right? And again, it's the fact that the affinity of the ligand to the receptor decreases substantially upon eversion that allows the molecule to move specifically in one direction, right? So here we're using the energy from ATP hydrolysis, whether we're talking about ABC transporters or P-type ATPases, to drive conformational changes that are moving molecules in one direction.
Like the reason it decreases the affinity, like what, like it does not such a different force, different acid?
Yeah, well, we have to consider, right, when we get the ligand binding here, well, what determines the interaction force, let's say, or the binding energy between a ligand and a receptor? What determines the binding energy between a ligand and a receptor? Right, all those favorable interactions, shape complementarity to ensure that we're forming favorable hydrogen bonds or ionic interactions or van der Waals interactions or anything like that. So this relies heavily on the shape of the active site or the binding site and on the shape of the molecule. Now what happens when we invert is now we basically destroy all of those interactions, right? We are going from here, right? We can imagine we have our ligand here all nice and happy making favorable weak interactions to this, right? So now all of those, the shape here, right, in the binding site has basically been destroyed, right? So all those favorable interactions are no longer present, so there's nothing really holding the ligand there. So the affinity is decreased. And the same is true for P-type ATPases, right? Notice this is on purpose. The structure here is very nicely attuned to hold on to calciums. Once we get that conformational change brought about by the phosphorylation of aspartate 351, well, now the shape changes drastically so that the calciums are not held strongly at all. All right, so now we get to something far more interesting, in my opinion, here, carriers, right? So carriers come in either active forms, we call this secondary active transport, or in passive transport form. So here in these secondary active transports, called active because we still require energy to move a molecule against its concentration gradient. Secondary because we're not using the primary source of energy that we normally use, which is ATP. Instead, we're using secondary energy, in this case, the energy from the concentration gradient of another molecule. So let's imagine the case here of what we call an anti-order. Here we're moving two molecules in opposite physical directions, right? But really what we're doing is we're using the energy of A to move B. So let's imagine that A has a high concentration here and a low concentration here. Well, A wants to move in this direction, right? That releases energy. That energy is captured by this antiporter and is used to move B from an area of low concentration to an area of high concentration. So it's capturing the energy released from allowing A to move down its concentration gradient in order to move B against its concentration gradient. Now a symporter is doing exactly the same thing. The only difference is the physical direction in which the two molecules are moving, right? So we have, in this case, A moving from high to low, so again, releasing energy. But in this case, B has an opposite concentration gradient than what it had here. It's low on the outside and high on the inside, right? So still we're using the energy of A moving down to move B against its concentration gradient. And here we're doing the same thing. It just so happens that the concentration gradient of B is reversed between antiporters and symporters, right? Antiporters because they're moving in opposite directions, symporters because they're moving in the same direction. But the principle is the same. They're doing exactly the same thing. They're capturing energy of the concentration gradient of A to move B against its concentration gradient. And antiporters are what we call essentially carriers, or in these facilitate facilitated diffusion. And so what is facilitated diffusion? Well, notice now we're moving A only, right? And the only thing that determines in which direction A is going to move is the concentration gradient and electrical gradient of A, right? A is higher on the outside and low on the inside, then we'll move down. If A is high on the inside and low on the outside, then we'll move up, right? That's it. But the reason we call it facilitated diffusion is because these uniporters are selective. So for example, when we talk about action potentials in the next few slides, right? Really what's going on is we're opening a hole in the membrane that is going to specifically allow, say, potassium through, or it's going to allow sodium through, or it's going to allow calcium through, right? It's not going to allow any ion through, so they're selective, and they allow diffusion to occur in a facilitated way. So they're facilitated diffusion. They're selective for their specific ions. So we have potassium channels, sodium channels, calcium channels, so on and so on. Additionally, these channels are almost always gated. Okay, they have a literal gate that either closes them or opens them. Right, they can be gated in a few different ways. We can have a voltage-gated channel, right, where the voltage of the cell itself actually either opens or closes the membrane, or we can have a ligand-gated channel, where the presence of some ligand, some other ligand, opens or closes the channel to a specific molecule. So for example, the acetylcholine receptor in the neurons, which we'll talk about today. The acetylcholine receptor is an example of a ligand-gated channel. In the presence of acetylcholine, the acetylcholine receptor opens and allows both sodium and potassium to move freely up and or down their concentration gradients. That's a ligand-gated channel. It's gated by the presence of acetylcholine. We can have a voltage-gated channel in the case of the sodium channel. In this case, the sodium channel opens, right, once the membrane potential reaches negative 40 millivolts, right? So when we go from negative 60 to negative 40, all right, well then that channel opens. So it's gated by voltage. We'll see how they work together. But here we have an example of a carrier, right? The lactose permease. And lactose permease is responsible for taking up lactose in bacterial cells. Okay? And we have to imagine, right, that we're a bacteria. We're in this hostile environment. There might not be a lot of food, so the concentration of carbohydrates and other fuel sources are pretty limited. All right, they're pretty scarce unless you're growing in a lab where you're nice and happy. That means the concentration of these carbohydrates, for example, lactose, is going to be higher on the inside of the cell than on the outside of the cell, right? And when a bacteria encounters a carbohydrate, it's just gonna immediately wanna take it up because it doesn't know the next time it's gonna encounter a carbohydrate. So how do we move that carbohydrate, in this case lactose, from the outside, which has scarce lactose, to the inside, which is rich in lactose? Oh, we need some energy to do that because we're going from an area of low concentration to an area of high concentration. As it turns out, the pH outside, right? The pH outside tends to be a little bit lower than the pH on the inside of the cell, right? So remember, biological neutral somewhere around 7.4, 7.5. So let's say that we are at actual neutral, pH 7. That means the concentration of protons on the outside is going to be a little bit higher than the concentration of protons on the inside. So protons want to move in, right? And lactose wants to move out. But if we want to move lactose in, well, why don't we allow a proton to move in? That's going to release some energy and happen spontaneously. We can capture that energy to move a lactose in against its concentration gradient. And that's what lactose permease does. This is an example of a syn-porter, right? Proton in, lactose in, they're all moving in the same direction. So what happens here is in the initial state, we have an exposed either aspartate or glutamate, something like that. This is going to be protonated to form either aspartic acid or glutamic acid. So now we have COOH, right? And this allows lactose to bind, right? The protonation of this aspartate or glutamate allows lactose to bind. In this state, once lactose binds, right? Well, now this changes the configuration, right? So we have an inversion event here, and this changes the affinity of the binding site for lactose. So lactose is released. Again, now the pH is higher on the inside of the cell, so this aspartate or glutamate is deprotonated, right? We get that proton in. Once we lose this proton, right, now we reverse the eversion and we are back to our initial state, right? So allowing the proton to move in is powering these eversion events, which also moves lactose into the cell. All right, now on to channels, right, which are critically important. They seem more boring because, oh, they're just letting ions through, but once you see how they're used in the cell, it's actually fascinating. All right, so first thing that we want to establish is that under normal conditions, the concentration of sodium inside the cell is quite low, and the concentration of sodium outside the cell is quite high. And that the concentration of potassium is reversed, right? So it's high on the inside and low on the outside. Okay, so we have energy stored in these gradients. Sodium wants to move in, and potassium wants to move out. And these concentration gradients were established by the P-type ATPase, the sodium-potassium pump, right? So in this case, what are we doing? We're just using ATP energy to move potassium in and to move sodium out. So the concentration of potassium is going to be high on the inside, as we see here, right? Potassium high on the outside, inside rather, and sodium is high on the outside. Again, this energy was established by hydrolyzing ATP by the P-type ATPase, the sodium-potassium pump. The next thing we have to understand is the resting membrane potential. And where does this come from? This comes from many different ions. It comes from obviously sodium and potassium, also from calcium and magnesium and all this. Okay? And what is the resting membrane potential? In other words, what is the delta V, right, at which the concentration gradient matches the electronic gradient, right? Why do we have to figure this out? Well, let's imagine that we have two compartments, okay? There's nothing about cells or anything like that right now. But we have two compartments made up of X plus, which are these blue guys here, on both sides. And on both sides, we have enough minus ions, doesn't matter what they are, there's enough anions to counteract the balance. So we have zero charge over here and zero charge over here. But on the left, we have more X plus and these anions. So the concentration of X plus is higher on this side. And on the right, we have fewer X plus and anions, right? So we have more X plus over here than we do over here, but we have zero charge on both sides. So at our initial state, what we have is only our concentration gradient. There is no reason for X plus to move one way or the other based on its electronic component of the delta G equation we went over before. Now let's open a hole, right? We're going to open a hole in such a way that it's only permeable to X plus, right? So what's going to happen in this case? Well, X plus will now begin to move out here, right? And what's gonna happen? Well, eventually, there's gonna be more X plus over here than the anions, minus, whatever, right? So we'll have more X plus than minus ions, so we'll start to build up a positive charge over here, which means eventually moving X plus over here is gonna get more difficult because we're trying to move a positively charged ion into an area that's already positively charged. So eventually we will reach some equilibrium, right? So what is that equilibrium? Well, it's when the concentration gradient equals the electronic gradient, right? So we can set this term equal to this term, solve for V, and what we get is the Nernst equation, negative RT over ZF times ln X in over X out, right? And what does this mean? That means that in our initial state, right, in our initial state, there is an electronic potential difference between these two. And if we allow X plus to move through that barrier, right, we will generate a voltage, right? And what is that voltage? It is VEQ, the equilibrium voltage, right? And we can see that the equilibrium voltage is dependent on the concentration of X on one side versus the other, all right? So if we have a large concentration gradient, that means that we will have a large VEQ. If we have a smaller concentration gradient, we'll have a smaller VEQ, and so on and so on. Okay? If we look at the typical concentrations of sodium and potassium, we can see that the VEQ for sodium, there's an example in your books if you want to prove it to yourself, the EQ voltage for sodium is 62 millivolts, and the EQ voltage for potassium is negative 98 millivolts. And what does this mean? That means that, let's say that we have a cell under normal conditions, right, and we poke a hole in it that is only permeable to Na+. Well, now what's going to happen is that the membrane potential of the cell will be equal to positive 62 millivolts, right? If we do the same thing for potassium, we poke a hole in that's permeable only to potassium, well now that shoots up, or rather down, to negative 98 millivolts, right? So when we have a channel that is permeable to a specific ion, the membrane potential shoots towards that particular VEQ for that enzyme, or for that ion rather, okay? And we can actually measure this in real time with a high degree of resolution using a method called patch plating. So essentially what we have is our cell, right? And we have a patch pipette. And essentially what this is a very expensive pipette, okay? And you attach it to the cell and you apply some suction. And what happens now is that any movement of ions, of course, is going to induce a current, right? We're moving electrons or positive charges either in or out of the pipette now. Inside the pipette, there's an electrode hooked up to an amplifier which can measure very small, I'm talking picoamps worth of current, right? So when a channel opens, right, the current drops, right? And when the channel closes, the the current is at its baseline here, sorry, right? So the current drops to negative, right, or it's at its baseline. So we can see here, all these, with a time resolution of, here you can see 400 milliseconds, you see exactly when specific channels are open or closed, and we can also measure the membrane potential overall in the cell attached mode, right? So we can measure the membrane potential here. And so what we see is that an action potential looks like this. Upon initiation of the action potential, first we get what we call depolarization, right? So we depolarize the membrane, followed by repolarization, okay, at the postsynaptic neuron. We'll see how this looks like in just a second. But before we start dissecting why an action potential looks like this and how an action potential travels through a neuron, let's look at the different components that mediate these action potentials. First, we have the sodium channel. And the sodium channel was first identified by purifying it using tetrodotoxin. Tetrodotoxin, right, does anyone know where we find it? Obviously in pufferfish, but you know why it's so dangerous, right? Why pufferfish is actually quite dangerous to eat if it's not prepared correctly. You watch that Simpsons episode? And date myself all over this year. All right. Tetrodotoxin is a sodium channel inhibitor. It blocks the sodium channel. And of course, the sodium channel we just mentioned is one of the channels that is responsible for generating this action potential. It binds very strongly to the sodium channel. Actually, as little as 10 nanograms of tetrodotoxin is lethal. And because it binds so well to the sodium channel, it's actually used in affinity chromatography to purify the sodium channels. Remember, we talked about affinity purification in chapter 3. In this case, this tetrodotoxin was immobilized onto the solid phase resin, and it was used to bind the sodium channel, right? So now we have all this sodium channel, the structure of the sodium channel was solved, and we see it is made up of 4 repeats, and each repeat looks very similar, right? So we have 5 hydrophobic domains, all right? Right, S1, S2, S3, S5, and S6. S4 is less hydrophobic, right? S4 is actually made up of mostly positively charged residues, such as lysine, right? And that's going to be important in just a minute. So we have these 4 repeats made up of 5 hydrophobic regions. And through hydrophobicity analysis, it was suspected that they probably are forming transmembrane alpha helices. We talked about that last week, right? So now we have these 5 transmembrane alpha helices, this one very positively charged alpha helix, and this repeats 4 times, right? When they isolated the calcium channel, they found basically the same structure, 4 repeats with 5 hydrophobic alpha helices, transmembrane, and a positively charged region here. So the positively charged alpha helix is always S4, and the other 5 are S1, 2, 3, 5, and 6. The potassium channel was more difficult to analyze, right? There was no strong inhibitor of the potassium channel like tetrodotoxin, so isolating it was quite different. It was actually isolated first from fruit flies. It's called shaker because one of the phenotypes that they found was that when the flies were anesthetized, I used to work with flies. The way you work with the flies, you have to immobilize them first, otherwise you just got fruit flies all over the place. You have to, and then you have to knock them out with carbon dioxide, and then just, go into a coma. We used to study the brains specifically, so you have to isolate the heads. The way you isolate the heads is even more gruesome. You just freeze the flies in dry ice, and you just bang them in a tube real hard until the heads just fall off. It's gnarly science sometimes. Don't get started on mice. Okay. These shaker potassium. So the way this is, I saw some poor graduate student when he was working through 20,000 vials of flies. He noticed that some of them, when he would knock them out with carbon dioxide, it would start to convulse, which is even more gruesome. And they found out that the mutation they had was in this potassium channel. And it turns out, when you sequence the potassium channel, you also find 5 hydrophobic alpha helices and 1 positively charged alpha helix. Okay, but you don't have the 4 repeats. But further studies showed that actually to form a functional channel, 4 individual polypeptides of this shaker channel come together to form this potassium channel. So now we have all these ion channels, and they are what? They are mainly made up of these repeats that have these hydrophobic alpha helices and this positively charged alpha helix. So this must be important in some way. And it was also known, right, that here, okay, when we are depolarizing and repolarizing the membrane, here what we're seeing is just an increased permeability to sodium, and here what we're seeing is increased permeability to potassium, right? This was measured separately. So there must be some voltage gating that allows this increased permeability to sodium once we reach a certain threshold. And once we reach a second threshold, there must be some mechanism to gate the channel that increases the permeability to potassium. So in this structure here of the sodium channel and the potassium channel, we had something that might approach that. Because we have these transmembrane alpha helices, right, S1, 2, 3, well, these are probably embedded in the membrane. S4, because it's mostly positively charged, would have the ability to respond to changes in membrane potential, all right, because of that charge. S5 and S6, right, these form the actual pore of the potassium or sodium channel, right? So these form the actual hole through which the ions flow. This was discovered by looking at the potassium channel bacteria. The potassium channel bacteria only have S5 and S6. There's no reason why a bacteria would need to have a voltage-gated channel. So the only thing that they require is the S5 and S6. By comparing the sequences, we can see, right, S5 and S6 probably make up the pore. Okay, so now we have these 6 alpha helices. Again, S4 is acting likely as the voltage sensor, and S1, 2, and 3 are embedded in the membrane. S5 and S6 are making up the pore. Now, when we look at the pore, S5 and S6, the question really was, how are they selective for either sodium or potassium? So if we look at the potassium channel, right, the bacterial potassium channel is because it's simpler, we don't have to worry about the gating or anything like that. How is the bacterial potassium channel specific to potassium, but not allow, for example, sodium through, right? Why do we expect sodium to make it through? Well, we can look at the ionic radius of these different ions, right? So here we have potassium right in the middle. Larger ions, such as rubidium or cesium, these are just physically too large to make it through the channel. So in this case, that's perfectly logical, all right? Larger ions can't make it through. But why wouldn't sodium be able to make it through, being about 10% smaller than potassium, right? How is that prohibited? Well, it turns out that we have to remember that in aqueous solution, we don't have all these just, positive or negative ions floating around. They're actually surrounded by a hydration shell, right? They're coordinated with 5 or 6 water molecules. In the case of potassium, we have 8 water molecules coordinated with the potassium. In the case of sodium, we have 6 water molecules coordinated with. Now, the radius of the ion plus the hydration shell is quite large, right? Even for sodium, it's too large to fit into the selectivity filter of our sodium pump, right? So what has to happen for either potassium or sodium to make it through the channel here? Sorry, not pump, channel. We have to remove all of these waters. So in the case of potassium, we have to remove all 8 of these waters. In the case of sodium, we have to remove all 6 of these waters. And that's, of course, going to require energy, right? We're breaking bonds, in this case, coordinated bonds. It's going to require energy. And that energy has to be paid by something. There's no energy here, right? This is not a pump, this is not an ATPase. So where is that energy coming from? Well, it's coming from the structure of the channel itself. The residues that line the channel are not random, they're specifically chosen so that they can replace all of the coordinated bonds that used to be taken up by water. So when we break these coordinated bonds with water, we replace them with non-covalent interactions in the channel itself. So the energy of desolvation is replaced with resolvation within the potassium channel, right? Paid for by the interactions of the residues lining that channel. The same is not true for sodium. Sodium still, we have to get rid of these 6 waters. That's our desolvation energy. But sodium is too small to make effective contact with all of the same residues, right? So the energy that we get back from resolvation is not enough to pay the energy of desolvation. So sodium is prohibited then from entering the channel because not enough energy is put back by the channel to repay getting rid of those 6 waters that were in coordination with the sodium, right? So now we can select for specific ions, either from excluding them because they're too big, or by very carefully selecting the residues that line these channels so that they only properly pay back the energy of desolvation with from the ion that they are supposed to be selecting for, right? So that's how smaller ions like lithium and sodium are prohibited from entering this channel. And what we see is that once we get sodium, flowing through the channel, what we see is that they basically flow as if the channel weren't there. You think, right, while you're breaking these interactions with the water and making interactions with the residues in the channel, so this is going to slow the sodium down, and what we see is that that's not actually the case. They flow as if the channel weren't even there. And the way that they do this is effectively just through electrostatic repulsion, right? We can imagine if we're moving down our concentration gradient, or have a high concentration of potassium or sodium or calcium, whatever, doesn't matter. We have a high concentration of potassium here, okay? low concentration here. So already we have our concentration gradient which wants it to move up, right? But again, why isn't it slowed down by all these interactions? Because the first sodium that binds is not going to be pushed up by all of these positively charged sodiums down here, right? Once we push the first sodium up, well, now this frees up another binding site here. This positive charge pushes up on this positive charge over here. This positive charge doesn't push down on this positive charge because it's also being pushed up by all these positive charges, right? So all of these positive charges are constantly pushing the sodiums up and up and up and up, so that the rate at which sodiums move through the channel approaches or is similar of that as if the channel weren't even there, as if we just poked a theoretical hole in the membrane. Now, how are these channels voltage gated? And again, there was suspicion that S4 was actually the voltage sensor of these either the sodium or potassium channels. Okay, and once the structure was solved for the potassium channel, it was confirmed that in fact, yes, the S5 and S6 parts of the repeat, they form the pore through which sodium passes through, right? So what we're looking at here is essentially, right, just S5 and S6, okay? S1, 2, and 3 are flat, and they're embedded in the membrane, right? So they're made up of mostly hydrophobic residues, so they're embedded in the membrane. S4 is only slightly embedded in the membrane and is positively charged, okay? Now, when we have positive charges on the inside, right? So here we have the inside of the cell, and here we have the outside of the cell, but we have more positive charge on the inside of the cell, right? Or a more positive membrane potential. This is going to push these S4 helices or S4 paddles up, right? And when these are pushed up, this is going to open the channel, okay? And the same is true for the sodium channel, right? They have basically the same structure. The only difference really is in the composition of their S5 and S6 helices, so that they can be specific for either sodium or potassium. I promise this is all going to come together in just a minute. All right, so what we have here then is this, right? Once we have a more positive membrane potential, again, the S4 paddle is pushed up, right, into the membrane, and this opens the channel and allows either sodium or potassium to move through. And this would be great, but if we go back, right, a few slides, well, we also need to repolarize the membrane. So we can't simply just open a sodium channel or potassium channel. If we open both of them, let's say, right, if we open both the sodium channel and potassium channel, the membrane potential would approach, what is this, 98 minus 60, 30, so about 15 volts, and then just stay there, right? That would be the new VM for this cell. But that's not what's happening here. We're repolarizing it. So what we need is a way to open these channels and then very quickly close those channels. Notice the time scale here is in milliseconds, right? So we need a way to open the channels and then close the channels to repolarize them. And the way that they're closed so quickly is through the use of this inactivation domain. And this was discovered actually kind of by accident. But basically, there was a mutant where the channel would open, right? But then it would stay open. There was no way that it would close, right? So we would have a situation where our membrane potential rose up and then flatlined because the channel never closed back again, so we could never reestablish that concentration gradient and that VM of negative 60 millivolts. But what they found was that at the N-terminus of this mutant potassium channel, they were missing, I don't know, like 15 or 20 amino acids, right? So they synthesized those 15 or 20 amino acids and they just threw it into the solution. What they found was that as soon as they added the peptide, right, the channel closed. And so the hypothesis with this, which later has been proven true, is that at the N-terminus of these voltage-gated channels, there is an inactivation domain, right? This has low affinity, or basically no affinity, for the channel when the channel is closed, but once the channel opens, it gains affinity for the channel, or rather the channel gains affinity for the inactivation domain. And this now homes into the open channel, which closes it, right? So the voltage sensing paddle is still holding the channel open, but the channel is now blocked by the inactivation domain. In fact, you can vary how long the channel stays open by varying how long this tail is that holds the inactivation domain. The longer the tail, the longer it stays open, because the longer it takes to home into the open channel. So now what we have to do is to reset this voltage sensor back to its off position. This will reduce the affinity of the open channel, because now it's closed, and then the inactivation domain will come back out, reopening the channel. How long it takes for that to happen, right? How long it takes for this inactivation domain to be released from the still open channel determines something that we call the refractory period, okay? And what that means is that once a channel has been opened, it's gonna take some amount of time before it can be opened again. That amount of time is determined by the amount of time it takes for this inactivation domain to be released, right? So as long as this inactivation domain is still here, this cannot be activated again. Again, that's the refractory period, how long it takes for this to be operable again. All right, so now we have our sodium channels, our potassium channels, and we see, we come back here to our membrane potential, action potential here. So we see what's going on, right? Well, first we're approaching a very positive number, and this would be potassium, or rather sodium, right? So sodium is positive 62 compared to our negative 60. So effectively, what's going on first is that we are increasing the permeability to sodium, and then at some point we are now no longer permeable to sodium. So the action potential goes back down negative towards potassium, right? So now we are no longer open to sodium and we're open to potassium, so we shoot back down towards negative 98. So we need a way to first be permeable to sodium, turn off sodium, then be permeable to potassium, then turn off potassium, and then reestablish negative 60. But we still don't know how we got here, right? What initiated this action potential? And the thing that initiates this action potential very commonly, almost always, is acetylcholine, the acetylcholine receptor. The acetylcholine receptor is essentially a channel that is equally permeable to both sodium and potassium, okay? And what does this mean? Well, if we open a channel that is equally permeable to both sodium and potassium, the membrane potential will approach the average of the VEQs for sodium and potassium, in this case about positive 15 volt or millivolts, okay? So we go from negative 60 to 15. Well, as it turns out, the threshold voltage for going from this to this in the sodium channel is about negative 40 millivolts. So we start at negative 60, we open both channels with the acetylcholine receptor, which we'll go over in a minute. We pass negative 40 on our way up to positive 15. At this point, we open the sodium channels. So now we're more permeable to sodium than the other potassium. So now we shoot way up to positive 68. But before we get there, the inactivation domain for the sodium channel homes. So now we're not permeable to sodium, but we are still permeable to potassium. So we shoot back down to negative 98, right? And we'll see all those in just a sec, all put together. But here we have the acetylcholine receptor. This is present on postsynaptic neurons. And essentially what happens is these bind acetylcholine, which is released by the presynaptic neuron. The acetylcholine binds to the interface between alpha and delta subunits, and between, or rather, alpha and gamma subunits, and between the alpha and delta subunits here, right? So acetylcholine binds here, and effectively what happens is that it opens the channel, right? So I don't want to get too bogged down into this. There's already been a lot of detail. So in the presence of acetylcholine, this channel opens, which is equally permeable to both sodium and potassium. This is what initiates the action potential. All right? And where does the acetylcholine come from? Well, this is what we call, or the junction between two neurons, the gap junction. At the end of the presynaptic neuron, so there's been a nerve impulse making its way down through this nerve cell. Once it reaches the end, this triggers the exocytosis of all of these acetylcholine-filled vesicles. So now we have all this acetylcholine being spilled out into the synaptic cleft, the area between the two neurons, right? And all this acetylcholine will then bind the acetylcholine receptor, right? Triggering sodium to come in and potassium to come out, right? This sends the voltage, membrane potential, up towards positive 15. On its way, it's passing. All right, so let's just go through the steps here, right? So here we're at resting potential. Here we get the initiation of an action potential from acetylcholine. So where we're gonna end up at is positive 15. But on our way up to positive 15, we pass negative 40 millivolts. That triggers the opening of both the sodium channel and the potassium channel. But the potassium channel opens more slowly due to the nature of its structure. So the sodium channel opens first, right? So at this point, after we're at negative 40, we are now more permeable to sodium than we are to potassium. So we shoot up towards positive 68, what's it, positive 62, somewhere around there? Yeah, positive 62. And before we get there, then the inactivation domain of the sodium channel homes and it turns it off. At the same time, right, so within this time frame, the acetylcholine in the synaptic cleft has been degraded by acetylcholinesterase, right? So now the acetylcholine receptor is no longer open. So at this point, we are now closing off the acetylcholine receptor and we're closing off the sodium receptor, or the sodium channel rather. The only thing that should be open at this point is the sodium channel. So our memory potential shoots back down towards negative 98. But before we get to negative 98, the inactivation domain of the potassium channel homes into the potassium channel pore, and so we get to begin to reestablish our negative 60 millivolts. And this propagates all the way down the neuron in one direction because of the refractory period. Right, so say we have our neuron here. Right, here we have the presynaptic neuron, right? So here we release acetylcholine, right? Let's just go through all the steps. Right, so we release acetylcholine, we're at minus 60, here we're at plus 40. Down here, right, the scale is really bad. Let's just go, it's minus 100, I know my scaling's off, please bear with me. All right, so now we shoot up towards positive, is this even right? No, whatever, minus 60. So we shoot up towards positive 15. Before we get there, we get to minus 40. So we open up our sodium channel, so we shoot up, right? Here we close off acetylcholine receptor, we close off the sodium channel, and we open the potassium channel. So we shoot that down towards negative 98. Before we get there, we close the potassium channel, right? So we shoot back up towards minus 60, right? And so this happens here, okay? Now when this happens here, it's gonna cause it to, it's gonna depolarize the membrane right next to it, which is gonna depolarize the membrane right next to it. So we're starting action potentials all the way down, right? So the direction is moving that way. But when we start our action potential, let's say here, right? Why is it only going to lead to an action potential over here and not over here, right? Well, it's because this had just been activated. So the refractory period is going to prevent it from starting an action potential back this way. So we're only able to move our action potential down in this direction. Right, so over here, right, once the signal is, say, down here, this region of the neuron might be ready to accept a new action potential, but now our action potential is so far away that it's not going to depolarize this region of the membrane, right? So we're moving our action potential only in one direction, all right? Whew. And so we can see what's happening right at each stage, right? So after about at time 0, all right, we get the opening of the sodium-potassium channel, the acetylcholine receptor. Then we get opening of the sodium channel. At the same time, we get opening of the potassium channel. But notice these are happening offset from each other because the potassium channel is slower to open, right? Then we get closing of the sodium channel after about 1 millisecond. At the same time, the potassium channel opens, right? And then again, we get closing of the potassium channel after about 2 milliseconds. Due to the homing of the inactivation domain, right? So here's our full our full process here. And so we start our action potential on one side of the neuron, right? And we propagate only in one direction, again, because the adjacent neurons in the wrong direction, not the adjacent neurons, the adjacent cell patches in the wrong direction are still in their refractory period, so they can initiate action potentials in the reverse direction, right? And many different, either naturally occurring toxins or synthetically made toxins trigger specifically components of these action potential propagation systems. So for example, what we have on the left here is cobra toxin. Right, so cobra toxin is actually used to isolate the acetylcholine receptor initially. Right? This cobra toxin binds to the acetylcholine receptor and it blocks the binding of acetylcholine. So it inhibits the opening of the sodium-potassium channel, right, the acetylcholine receptor, and so initiation of the action potential cannot proceed, right? So we don't even get initiation of the action potential because now these acetylcholine receptors are insensitive to acetylcholine. So cobra toxin, right, is a neurotoxin because it affects specifically transmission of these action potentials. This is a naturally occurring peptide, right, which is produced by a certain genus of snakes, right, cobras, cobra toxin. We can have man-made inhibitors also of action potentials. My favorite to talk about is sarin. So in the 1980s and 1990s in Japan, there was a cult that formed out of a yoga group called Aum Shinrikyo. Anyone heard of this? Huh? No? So basically, it was this guy that set up a yoga group, and then it just got really out of control. It was wild. So they killed the family of the guy that started it, and then basically they tried running for office. They started making drugs and different things. Basically became a gang, but they were really a cult. And they heard that they were going to get raided by the police, and instead of running or anything like that, they decided they were just going to distract the police by performing a biological terror attack on the Tokyo subway system. And they manufactured for themselves on a farm just a bunch of sarin and released it in the Tokyo subway. Ended up killing like 13 people and injuring 500. I think a couple of people went blind. All because they just got really out of control. Anyway, how does sarin work? It inhibits the acetylcholinesterase. Right, so acetylcholinesterase, yes.
Sorry, just one question.
Is
So
it
they
about
made it on
Shinrikyo?
they made it on a farm?
Oh yeah, they just made it. They
Okay, cool.
synthesized themselves.
Oh, hell yeah.
Yeah, they just were wild like that. I mean, they were terrible people. A bunch of documentaries. I highly recommend the last podcast on the last series about Shinrikyo was wild. Anyway, sorry. It's what we call a suicide inhibitor, right? So the acetylcholinesterase enzyme, which is responsible for turning off the acetylcholine receptor, is present in the synaptic cleft. And it breaks down, it hydrolyzes acetylcholine into acetate and choline. And this happens very quickly, right? So it happens right about here, it happens, okay? So that way, the channel is also the only way that we're altering the membrane potential now is by increasing the permeability to either sodium or potassium via their respective channels. But that can't happen if acetylcholinesterase doesn't degrade the acetylcholine very quickly. And that's what sarin does. Sarin covalently and non-enzymatically interacts with serine at position 200 in the acetylcholinesterase. Okay? And this serine is what's responsible for actually hydrolyzing acetylcholine. It turns it into this adduct here, which is inactive, right? Notice this serine no longer has the hydroxyl, which is critical for hydrolyzing that acetylcholine. This now is inactive, and so what happens, right? Well, what would we expect to happen in the presence of sarin? Well, the acetylcholine receptor becomes open. We approach positive 15, right? We have passed minus 40, and we've passed, and so both the sodium and potassium channels become open, right? but very quickly they're inactivated, right? So really what we have open is the acetylcholine receptor. So really what happens is we come up here to -15, we might get a little wiggle here from the sodium and potassium channels opening and closing, but then we flatline at +15 volts. And this, of course, is not an action potential. We need that repolarization to move that action potential down the membrane, right? So this serine, this acetylcholinesterase inhibitor, prevents the propagation of the action potential, whereas cobratoxin prevents the initiation of that action potential. But the effect is very much the same. Now these signals cannot travel to or from the neurons or the brain, and so this leads to respiratory failure, blindness, other things. Right? Now we can reverse this. There are antidotes to both of these. There's an inhibitor that can compete for binding there of the acetylcholine receptor. Here, acetylcholinesterase, once it's been inactivated, right, we can add pralidoxime, 2-PAM, right? 2-PAM, again, non-enzymatically interacts not with wild-type acetylcholinesterase, but instead with the inactivated adduct, right? And what we form is reactivated acetylcholinesterase. This is go on and be happy and do its thing. But we form this compound here, which is spontaneously hydrolyzed into phosphonate, this here, and 2-PAM, which is what we started with, right? So this non-enzymatically and importantly catalytically restores the activity of inactivated adduct here, all right? So a very small amount of antidote 2-PAM is successful in very rapidly restoring acetylcholinesterase activity. Problem is that you need to administer it before you die, right? It's not always possible. Right. All right, these are more inhibitors here of action potentials. I won't get too much into those. I'll leave you with those grim stories to investigate, and I will see you all on Friday. All right?